Is starting. Oh, let's make sure. Wait a minute. You're not kidding. Oh, we're good. It logs it out if there's nobody else is logged on. That's one of the problems with that. Nevertheless, we are now recording. So, if this is Micro 237D, Section D. I do Section E at 2 o'clock this afternoon, and more or less we'll try to do the same material for each group, and we'll just go from there. For those that have not had me yet, I'm Dr. Stein. Uh, Normally by this time, particularly for the students who had A and P1, normally I used to do a great deal of those sections. It's just the way there's been some transitions into who's teaching what in our department. That Dr. Robbins had most of the sections uh, this time, and I see some of the students here from our AP2 section that we had in the fall. Nevertheless, that's the introductory nugget, and welcome for today. So, without further ado, me sitting here and reading this syllabus to you is unnecessary. I'll just do the highlight film version because we don't need to do much more. This is it. We're here. I like the room, and I'm in this room for Section E and for another class that I'm teaching. It's a big lecture day every Monday, Wednesday, and Friday. The usual boilerplate material. Typically, and I don't know what your schedules are, but when I'm on campus between 9 and 10 to 12 to 1 on Monday, Wednesdays, and Fridays, that's when I'm most readily available. I typically am in my office over in the main portion of John Jay and 218. You all become, if you're taking micro, you become very familiar with that part of the building because you have micro lab over there. Okay. And so the lab is sort of when you walk in that main door, making a left and making a right, you head to the lab. But if you go straight in through the doors that take you really into the offices section, I'm in the room what we call the atrium. You enter here is at the second floor, and I'm in the far corner to the right, not quite the corner office. If you're looking for me, I'm there. You're welcome anytime. But if we need something significant to discuss, make an appointment, and we can log in and do them virtually easily enough. You can read the objectives as well as I can. The focal point for our micro is certainly bacteria and viruses. Okay. It doesn't preclude that we're not going to look at other organisms, but that's surely the heart of this microbiology. The lab is a bacteria lab, meaning you deal with bacteria more than anything else. Maybe there's going to be some yeast and fungi that you'll be working with as well. Those are in a slightly different category, and we'll get to all the categories and things like that are there. But principally, our focal point from lecture is understanding the ramifications of those. And my focus point, and inevitably, I have the better part of 50 students for micro in these two sections. You always have to teach to basically your target audience, and the vast majority are nursing majors. If not nursing majors, then you're in a health-related field like nuclear imaging, etc. Those kinds of fields. That's where the vast majority. There's some that are not, okay, for different reasons that are there, and that's great. So I mean, because you get a broader range of people to communicate with about that. But so my focus point for this, and the way we have framed this particular microbiology, is oriented toward those students, and my orientation is very much clinical, meaning that I want to give it to you from a perspective of how it affects people, how you go about treating it, what the problems are particularly, so that and for many of you, if you're going to go into a health-related field, that you understand what the ramifications are. If somebody walks, if you're doing a procedure on a patient with influenza or RSV, you know what to do to be able to treat them, to make sure that they don't develop further problems, and you don't expose yourself, or more importantly, someone else in the hospital to a particular problem. So a lot of that is what we cover. Those are the clinical ramifications and the clinical aspects. So I like to frame it roughly. The first half is you got to go through the basic bacteria and viral anatomy and physiology. Okay, and it's different because you're not used to it. They're not the same thing that we are. Most of your frame from the time you have started to study is dealing with what we call the eukaryotic cell, our cell, the A and P cell, the nucleus and the ribosomes and the mitochondria and all that good stuff, right? membrane-bound organelles. You heard that forever in AP1. We have plenty of those. Yeast are in that category. Protozoans are in that category. The single most deadly disease in sub-Saharan Africa, a major giant continent, is a disease from that category, malaria. It's a protozoan, a eukaryotic pathogen. Disease-causing organism that's very similar to our cells. Why is that a problem? Because if we try to treat it and kill those cells, we're going to harm our cells. There's a lot of ramifications with regard to that. But bacteria are different. Bacteria are a single-celled organism with a cell wall that is very different internally, though it's a living thing. And viruses aren't living at all, by any definition. Living things have to have what? You all learned about this typically in A&P1 and previously. They have to have DNA, they have to have RNA, they have to be able to do energy transformations, gobbling up stuff, creating energy, getting rid of waste products, replicating itself, blah, 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 blah. All that stuff, boundaries, responding to environmental stressors, et cetera, et cetera. That's what living things do, okay? Viruses don't meet that standard. One, because they're not made out of cells. And our definition of cells in cell theory is everything has to be made out of cells, either a single cell or multiple cells. And all cells come from previous cells, I guess, other than the first one. That's it. Viruses don't apply to that. They're totally different. They're acellular forms, and they're not the only ones. So we'll be looking at that as well. So there's a lot, and that's, I think, the difficulty and why, from a science perspective, from the biology sphere here, particularly for nursing students, why this is typically a fairly difficult class. Because it's a difficult term and again, I'm speaking to mostly the entry-level nursing students. Look what you got to deal with. AP2, which is more about physiology and a little bit less about anatomy, and more about things that can go wrong when you dealt with the basic structure in AP1. And you're dealing with both lecture courses with labs, science-based labs, and then you have to start getting ready for all that nursing stuff that goes on. Ugh. Whatever core courses you're stuck taking. Speech 1000, speech 1050. So I have a fair amount of experience doing that. So that's basically what we're trying to do. You can read through that as well as I can. I don't know how they're doing the text. I don't sell textbooks. They've got book bundles and all that. Whatever you're going to do. If you want to buy a text, buy a text. If you don't want to buy a text, don't buy a text. The oddball, and I emailed you about that, is the Connect package and access to it. I have the information if you wish to log on to that. There are professors who I know that they use that mastering content for A&P, which I don't particularly use. I mean, that's fine. I mean, however they want to do it, that's what it is. I don't use this, but I'm happy with people. I think the software used to be free. Let's put it that way. It's not anymore. That shouldn't surprise anybody. There's a cost associated with it. So we used to be able to access animations and little work things and question modules freely, but they don't have that now. So that's where that. So if you want to use it, that's fine. I created a site. If I didn't send it to you, it's easy for me to find the login information to send to your students. I think I did send it to you, et cetera, et cetera, et cetera. And with the whole bookstore thing not being a book, the traditional model of the bookstore being mostly, and I understand that most of you, I mean, I'll have to look in the room. Nearly everybody's got a laptop open or a pad or some type of technology. Gone are the days of the typical, the written notes, the handwritten type of stuff, but that's fine. We move forward, not backward. Standard grading, that's there. And if you haven't had me before, when it comes to lecture, I am exam-based, and there's typically 4. They are announced. Yes, you're going to have a study guide. They're going to be objective in format, meaning, true, false, multiple choice, matching, ta-da. Okay, standard stuff. No short answers. No way. I'll leave that for the lab part of it. Why do I do it that way? The class I come to from this, which we had downstairs, is MCAT preparation. medical college admissions test for the future students here wish to be doctors, particularly of some discipline, typically medical or osteopathic physicians. They take this particular test called the MCAT. It is an objective test. And what do you have to take? Again, speaking to most of you who are in a licensed field, and particularly the nurses, the NCLEX. And nothing matters if you don't pass that test. You can't get if you can't pass a licensure exam, then all of this was for naught. Okay, do something else. Those tests are objective. It's you versus the computer. There's no way to cheat. You're in an environment where if you are not familiar with how they administer these tests, you go to a place, you go through metal detectors. If you beep at all, they're going to hand wand you. You can't take a phone in. They'll give you a pencil and some scratch paper, and it's you versus the computer, and there's no way to look at anybody else's. And you'll answer X number of questions until they say you're done. And you don't know whether you're done because you answered them right or you answered them wrong. That's how it works. So that's when I hear about, and we get stories about cheating all the time. You're only cheating yourself. But sooner or later, it's going to be you versus the computer. And if you don't know it by now, then the thousands of dollars either mom and dad have spent or you're indebted for are gone. My one kid did. Get a commercial driver's license with a hazmat endorsement, drive a truck, and make $7,500, $1,000 a year. It's okay. He makes more money than I do. Better looking and younger. If you're going to the car show at the convention center this coming weekend, he got like an old, like basically empty old Mazda, either 7 or 8, and restored it that he's showing you. That's his hobby, is rebuilding cars. Nevertheless. Okay, so it's about the exams, and you'll have a study guide, and they are difficult. Okay, by design. You all you do is ask somebody who's had me before in any type of a class. I make the exams difficult because it's important for you to see the degree of difficulty that you might encounter, and I will typically grade adjust or scale curve, whatever you want to use, so that because I have an idea. So when someone gets 80%, usually they're going to probably 85% probably going to be an 8 on one of my tests. So don't look at absolute scores. Students always do that, so they're by design. I have to give you a degree of difficulty in order to be able to manage that. More about that down the road when we get to the exam stuff. That's it. All the rest of the stuff is pretty basic. Show up for the exams. Nice for you to show up here. I like to see smiling faces. If you haven't heard it by now, if you go to an instructor and they can't recognize you, don't have to know your name. But if they can't recognize your smiling face and you're going in, 6 weeks or 8 weeks in semester, oh my God, I'm not doing well. What can I do to help? They're not going to be very reasonable towards you. Let them see your bright face. We call it osmosis. Just sit there. Stein's rule number 1: show up if you can. If you can, give me a heads up. Within either before or shortly thereafter. We're recording this on the virtual site. You get to hear all the bad jokes for those who haven't before. Once again, shamelessly rehearsed in front of a mirror all the time. No new material is coming your way. Okay, at least we made the playoffs. No, they didn't win, but I'm okay. You're probably a Redskins fan. Thank God. Jaguars? Mom was in Jaguars. Just say. So you can see what I was pointing to here is you can see all this stuff is being recorded, and we will get there. Why is it not showing the board? Maybe it is. I don't know. Oh, I'm still on because I wasn't showing that particular content. I'm going back. And the rest of this is all generic material you don't have to know about. This is roughly the schedule. We're going to get started shortly. I hate to take more than 20 minutes. This is all the stuff, and most of these are with these different statements and things like that. This was a new one. You've probably seen it on your syllabi because this class has started yesterday. That these are required statements for either online or on-site courses, and we've qualified for both, all the different things, all the COVID policy and masks. Oh my God, let's not go through that again. I mean, that's why I don't know how. I think we won't see the ramifications of what went on, even though we're talking about COVID and related problems a lot here, of what it meant for you to folks to be, many of you, not in classrooms or tutoring remotely for a substantial amount of your education, either in secondary education or collegiate education. So I'm really curious about that. I was a very hard time, all that, and we'll more about that as time goes on. That's the intro stuff. We get started. Good. Back to Blackboard. Where was I? So I just reload my old Blackboard from last time I taught this, which was last spring. This is the textbook. These are the PowerPoints. I've outlined some of the chapters. I have some written notes. We were doing this a lot when we were doing COVID, and I was doing this from home. And I've just added those historical figures. These are my previous lectures. We used to use the Nestor textbook, which I will go to because I'm very comfortable with its illustrations. So I'll make you aware when I'm doing this. Like, for instance, we talk about Pasteur's famous experiment. I like that illustration better than one that Cowan has, but that's okay. Okay, and I have some animations. And so typically week 1 applies to exam 1, week 2 applies to exam 2, week 3 applies to exam 3, etc. You get the idea. And all these different things, like this is when we're going to start talking about gram-positive versus gram-negative cells, an extraordinarily important part. And I'll highlight to you what are the important things. Okay, good to go. And so, and this is kind of the stuff I've been using this outline, I don't know, for 20 years. So, and more or less, for the first chapter, we'll try to get through all this material. And every so often, I'll flip back to it, not in any specific order, because different textbooks use different ways of going through, but they all basically cover this material. So these are just some of the things we'll talk about. And we'll go from there. I don't prepare what I'm going to say so much as come up with what say, and I try to relate it to topical things as much as possible, shamelessly showing you pictures of my children and my pets, particularly. The cat has no idea what to do. He's a cat that likes to go out, and he's indignant. He goes to the door like at 6 in the morning, and it's freezing, and he's like, I'm not going out. And then a minute later, he's back at the door, and when I open it, he goes, looks at me like I haven't changed the weather yet. I haven't turned the temperature up. The cat has no idea. And the dog is just totally perplexed.
Does he not like the snow?
Oh, he's okay with the snow.
Oh.
It's funny, he doesn't like his feet wet in the rain. That bothers him.
So he doesn't, okay.
But
Maybe
the snow is okay.
Boots.
Snow is okay. We should get him boots.
That'd be so cute.
Yeah, he's cute enough as it is.
Boots would...
75 pounds of dog is a lot of dog. Down there, you can see the picture. A lot of dog. Soon to be 3. PowerPoint. Here we go. There we go. So Cowan, the way Cowan does her books, and whoever has put it together. For a quick word about PowerPoints. Don't come and tell any professor, oh, it said so on the PowerPoint. PowerPoints are not subject to editorial review. They hire a graphic artist, someone who makes PowerPoints to make PowerPoints. Textbooks are subject to editorial review, particularly the ones that we use. Meaning that you'll get mistakes and typos on PowerPoints all the time. So and this is all the stuff, the different kinds, what they do, a certain amount of science, because there's a lot in this about, when it comes to microorganisms, there's a lot about evolution. There's a lot about cell theory. There's a lot that goes on with this. And we do this constantly. Every time you hear the word, the letters GMO, we're dealing with bacteria and yeast, and we are genetically modifying. We're altering their DNA by inserting other DNA and effectively slaving them to make things for us. So that we do this all the time, both for good and unfortunately for bad. So just want to give you an idea. what diseases do to us, the different kinds, where there's a certain amount of this is more the anatomy and physiology part of the program. Acellular infectious agents, not living things, viruses, and something kind of neat that are called prions or prions, depending on how you pronounce them. I call them prions. Those are misfolded proteins that cause other proteins to misfold. The best example of what we know that plays a significant role in mad cow disease. More about that as the time goes on. And we'll look at the sizes and different things like that. So we're talking, it's a misnomer. Microbiology is inherently a misnomer. The ology means study of, the bio means life. So it's ostensibly we're studying living things that are too small to be seen with the naked eye. That's effectively the definition of microbiology, but it's a misnomer for what I just discussed. Viruses and prions and something called viroids, that all things that we know of, by definition, are not living things because they're not made out of cells. So these guys down at the bottom, they're not in the family. But bacteria, something we don't see in our environment, but we study, or at least learn a little bit about, and they're very interesting from a research perspective, are called archaea. So archaea from archaeology, if you want. They're the oldest of the organ, and interestingly, they are probably the ones that are a little bit closer to us. Surprises a lot of people. But then and we'll get to what it has to do with domain theories and different classifications. Protozoa, those are typically things you remember in high school when you got pond water and took a look at that, all those neat things that were spinning around in it, running around, amoebas kind of crying out everything. Those are protozoans, single-celled organisms that don't have a cell wall. And interestingly enough, they play a significant role in disease as well as in other things. Major problems with certain kinds of diseases, fungi and yeasts, something that play a role in diseases and play a role in daily life and are used as well as bacteria for genetic modification. Helminths are worms. I don't do a lot on worms for two reasons. One, typically they're only microbes, meaning microscopic entities, until they start to chain together, and then they're macroscopic. We can see them. And so to me, they fall more into the macroscopic than the microscopic word. But most importantly, I don't know enough about it to tell you about it. Okay, if you want to know about worms, you have an animal or you know a veterinarian, talk to them, because they're much more significant in understanding. I mean, I'll give you some information about it. Algae are effectively microscopic plants because they have chlorophyll. So they're much more closely aligned to the plant population, let's say, because of the presence of chlorophyll, than, say, bacteria that are there. And we'll talk a little, because they do certainly impact our lives. And again, the viruses and the prions not being part of the quote unquote realm of. So a better way to phrase that would say microbes, which implies all microscopic entities. Just giving you some sort of generalized information here. So I don't know that they're necessarily that easy to study. I think that's probably a misnomer. But here's the big problem. Why do we have so many problems with viruses? Why do we have so many problems with bacteria? Because they replicate. And that's what they're geared to do. They don't replicate the way our cells do. Okay? They don't undergo a process, by and large, like mitosis. Things like algae and protozoans do, because they're similar to our cells. But the whole ball game is, how do they beat us with vast numbers? If you don't address a virus or a bacterial infection early, big problem. They start to overwhelm right away. And the weaker your immune system is, something that many of you will be studying in AP2, the more difficult it is to get a handle on fighting these. So we are able, the good news, that's the bad news for sure. The good news about reproducing rapidly, and you'll see in the laboratory, is they're easy to study. we can grow a lot of them and take a look at how to attack them, whether by heat, whether by chemicals, whether by other kinds of manipulation. And those are some of the experiments that many of you will be doing in the laboratory. And you'll like your lab instructor for the most part, because the one I know is Dr. Dress, and he's great. And you'll have a good time with him. We can't see them, so we have to use microscopy to be able to see them. So we have to analyze them indirectly. And again, this is more lab than it is lecture. We look at what they do. Do they ferment certain sugars? And when you ferment something, typically you take a sugar and you make it into something acidic. Okay? And then you use indicator dyes to be able to see it. And we'll talk a little bit about microscopes, though that again, you're more about that in the laboratory. So which is not considered a microbe? By definition, a mushroom is even though it is in the family of yeasts and fungi, it's macroscopic in nature and very tasty. For those that don't know me, I use a lot of food analogies because, I have an existential relationship with food. I eat, therefore I am. Dr. Badger, our department chair, always has candy in his office, and today, yeah. So I won't be a week from yesterday, I won't be as happy because I have my annual physical. I'm sure he's going to yell at me. Sorry. So, without getting into the idea of evolution and ecology, and I don't want to make that into a discussion. From a science perspective, based on carbon dating, we've been around, and the Earth has been around for billions of years, and microbes were really the first things that we can classify as living. So, based on their studies scientifically, despite my age, I was not there for this. Close. I was telling my MCAT class that I took those examinations 51 years ago. So it has been, it wasn't 3.8 billion like many of them think. So they appeared then, and our belief is that everything stemmed from these first onset, this first sort of confluence of living things that occurred associated really with the presence of water being there and particles that somehow came together and formed something living. If you want, whatever your beliefs are, that's sort of the scientific aspect. And not all of them had the nucleus. If anything, the concept of having a nucleus came later, not earlier. The earliest organisms were not eukaryotic, but the name that we talk about is pro, for pre or pro for before. So pre-nuclear. EU typically means true or with. And the carrion is considered the nuclear or sort of complex center of development or control center for these organisms. So when you hear me use the term eukarya, it implies that they're very similar to our cells. They are indeed our cells from a standpoint that they have a nucleus. And what defines them more than anything else is membrane-bound organelles. We'll get to why our ideas they formed after by something called endosymbiotic theory, which is an important part of this class as well. So bacteria do not have membrane-bound organelles. Their organelles are within their cytoplasm, but they do not have a membrane. But the idea of what you did in AP1 is having that membrane imparts, or sort of implies, rather than say imparts, the idea that things can freely move in and out, but somewhat selectively. Stuff, we can bring materials into our nucleus. We can generate DNA, we can generate RNA. We send the RNA out of the nucleus to something, this is all just rehashing A and P, called a ribosome to make proteins, and then the proteins begin to make other structures. That doesn't happen. It happens differently in a bacteria. Bacteria have a confluence of DNA. We call it a nucleoid because it's like a nucleus, but it's not. Why is it not? It doesn't have membrane-bound organelles. And moreover, the DNA is configured differently. That's a big deal. Everybody knows our DNA, and you all learned about this, is in pairs. That whole 23andMe thing, right? 23 pairs, we got the XY and 22 others, okay, cool, I like that. Not so with bacteria. They have a big circle of DNA. That's all it is, a giant circle of DNA. And that circle is twisted and folded complexly to make something held together, something called a nucleoid, but there's no membrane around it. So that's important. Archaea are similar, but there's differences in their structure that we will get to. Okay. And so I've never used the term eukaryotes. A would again mean without. So this is the early idea. This is what we see today. This is based on the domain. You'll notice what was absent before when we talked about microorganisms, animals and plants. By definition, animals and plants are macroorganisms. You see them with the naked eye. They are composed of different eukaryotic cells. So they're not necessarily immediately in this construct until we get up to here. So we talk about whatever the common ancestor was. We have bacteria and archaea, and there's a lot. What's the major distinction between bacteria and archaea or archaea? How you want to pronounce it? Where they live. There's other differences. Archaea lived, if you think of what the planet was like when it was born, it was an extraordinarily harsh environment. Okay, so where do we see archaeal organisms? We see them in harsh environmental conditions. Bottom of the ocean, polar ice caps, thermal vents, volcanic caldera, all those types of things. So we study them because they have survived in harsh environments, and you get a lot of research from that. And there's a lot of interest in seeing those. Bacteria live basically everywhere. They even live in some of those harsher environments. But by and large, we're going to be looking at ones that are classified as either gram positive or gram negative. And we'll go over some of these other ones that are there. And when we start to talk about what made our cells, eukaryotic cells, without going crazy about it, we basically classify them from a microorganismal standpoint as how they get around. Do they basically, are they motivated by flagella, which a lot of them are? Are they motivated by cilia? Do they sort of move amoebically, sort of scooching around like a baby would scooch around? We classify them like that. And once they begin to group together, then we can see them with the naked eye, and they're no longer in the microbial aspects. So bacteria, to summarize, bacteria and archaea are single cells. Not predominantly, they're single cells. Eukarya, a lot are single cell. When we talk about those protozoans, those things in the water, by definition, they're single cell. They may group together, but they don't. It's not like our having multiple cells making up tissue that you learned about in anatomy and physiology. Okay, they're not making up by a larger structure. Eukaryotes, when they do group together, become by definition multicellular and visible. Okay, and there's a whole lot less of them than there are bacteria and archaea, and that's part of the problem. We are very misunderstood about bacteria. By and large, they're our friends. Oh my God, so-and-so has a staph infection. Most bacteria are occupying sites on our bodies that prevent bad guys from occupying those sites. They're within our bodies in the sense that they're within the alimentary canal in our gut. They're essential for it to function. They're essential to ferment things. They're essential to make a sufficient amount of different kinds of vitamins that are there. There are major protection against various kinds of diseases. Yeast, which are, and the most famous of all of these yeasts is called Candida albicans, is something that's located and would overwhelm in the mouth or rectally or vaginally, okay, but doesn't do so because of the presence of other organisms to suppress it. When that's not there, we get disease, and we'll talk down the road about things like vaginal yeast infections, which is your problem, or one that we get in the mouth called thrush, which many of you will see, which I get occasionally because of the medicines I take. So now that we're kind of giving you sort of an evolutionary timeline at the start, wherever this common ancestor was, and first, and I think the archaea came first, but maybe they're contemporaneous, not a big deal. Eukaryotic cells, then insects, then reptiles, the development of mammals, and us. We haven't been around that long. Okay. So the first concept is something you do in the laboratory. It's the idea of ubiquity. They're everywhere. It doesn't matter how many times you wash your hands or how many times you go over and hit that hand sanitizer thing. You know what it's like? You see one. You gotta go buy it. When did they see it? It's like an attraction. The fact that you just did that doesn't mean that you're now magically, your hands are sterile. We can't sterilize living things. It doesn't work that way. We do a whole unit on disinfection and sterilization. Okay, or you can make them, is relatively speaking, clean. In an operating room, when you wash your hands preparing for surgery, and I've only scrubbed in, I don't know, 1,000 operations, okay, more or less. All you're making do is you're making your hands clean enough so you can pick up what is going to be a sterile glove on the interior portion that's going to be in your hands. That's what you're doing. Not a thing a whole lot else. So they are everywhere. They're on the table. They're in this cup. They're on this keyboard. They're everywhere you touch. They're covering 100% of your skin 100% of the time. No matter whether you just got out of the shower, whether you hand sanitized your way on the way to work or on the way to the office, they're there. So they're everywhere. Whether they're deep inside, whether they're in hostile environments, whether they're inside of us, whatever your definition of that is, anywhere. And yes, they are essential to life. If they would disappear, more or less we would disappear. So all of this comes from a variety. When we talk about theories, scientifically, a theory is pretty much fact. Okay? It's almost like Newtonian laws. They've been around so long without anybody poking holes in them that they've reached. So theories are just about the way of laws. So when you hear about cell theories and theory of evolution or endosymbiotic theory, they're there. It's a little bit different than somebody said, I have a theory about why the Steelers didn't win that playoff game. Okay. That's more of you. That's not the same as a scientific theory. So the accumulation of changes that occur in organisms, they adapt to the environment. Okay. So interestingly, if we go back and revisit what Darwin did, and I'm not an evolutionary biologist, I'm not an expert in evolution, you can poke holes in what Darwin did. But basically his premise was right. It was called descent with modification. So as time goes on, basically we continually, all species, all living things continually mutate. And we'll go into this here in the class. We'll talk about errors that occur genetically. Every so often we don't capture one of those errors, and sometimes it makes that organism better able to survive. Organisms that can survive, particularly in hostile or changing environments, are more likely to mate with and reproduce. with other similar organisms. And eventually that change becomes incorporated. That's Darwin 101, descent with modification. So as we're able to modify and adapt to changing environments that are there. I mean, this is not a pleasant environment out there today. So, but we are able to adapt to it sort of indirectly. We put different clothing on, we protect exposed parts of things like that, right? We know how to do that. Bacteria don't have that luxury per se. But the fact that they have a cell wall protects them to a certain extent. Cold doesn't kill microorganisms. Cold stops them from growing. You have a perfect example of that at home or wherever you live. You have a refrigerator. You have a freezer. All it does, why does a refrigerator work when you put something in there? You limit the likelihood of something becoming spoiled by organisms developing that would cause spoilage in them. It's not forever. We've all seen that. I mean, how many times have you gone into your refrigerator or your freezer and go, this is a little shady looking. You left it in there too long. You did it the other day. You know, my wife gave me a certain set of instructions. You will now clean out the refrigerator. And of course, I knew exactly how to answer her. You've heard this before. I looked her right in the eye and I said, yes, dear. Right, which is why I am here for nearly 73 years. I know how to. This was an opportunity that many of you just missed. None of you, all of you sat there and went, yeah. You couldn't, nobody could have, you couldn't be 70, nearly 73 years old. Thanks. And I'm looking at you, Grace. This was your shot.
I know.
It's okay. When in doubt, we blame it on Grace. You just like, hey, gotta pick on somebody every day. What?
I said you're in your 40s.
Thank God. Somebody said, not in the 40s. Not the way I feel in the morning. Oh, my God, I got to get up and do it. Cat starts to me at 5 o'clock in the morning. I'm like, oh, I'm getting there. Just let me throw something on. I'll try to open the blankety blank door for you.
Whoa.
What? It's the cat.
I know. Do you love the cat?
I love the cat. Don't know why.
But you do. Do you
The
not
dog's a
love
little better. He's, I don't want to go out in this stuff. Okay, so other things, years and years of testing, well studied, well established, not a guess. Okay, fine. So yeah, it's widely accepted among scientists because it still remains unproven. No, I don't know about that. Okay, different things. They're talking about here. I don't know why they put this in photosynthesis. Something that without it, just to give you an idea of the importance of microorganisms, why I said before, if they weren't there, we wouldn't be here. Our oxygen supply comes from photosynthesis. Basically, we take inanimate things, carbon dioxide and water, and in the presence of photosynthetic organisms like plants, they are able to convert them to carbohydrate substances, and as a side product, it's really good, oxygen. Our planet is approximately 5/6 water. Most of the oxygen in our planet wide comes from photosynthetic organisms called, okay, that are in that category of microorganisms, chlorophyll containing either bacteria or algae. That's where now you and I, our oxygen we get, for the most part, comes from the photosynthetic organisms that are still here, that are land-based. But planet-wide, it is mostly from algae, because that's what's in the ocean. That's where most of the oxygen comes from. Okay, so formation of oxygen, photosynthesis before was different. Okay, but we are effectively being able to utilize sunlight, create edible substances. As an aside benefit, we get oxygen. So photosynthetic microbes are responsible for 70% of the Earth's photosynthesis. microorganisms like algae, photosynthetic bacteria that are there. Without them, we would not be able to do this, to be able to produce not only carbon dioxide, nitric oxide, methane, all of which play an important role. So when we are able to talk about carbon fixation, that's being able to take inorganic carbon dioxide from the environment and basically staple those molecules together so we can make, as a start, carbohydrates and other carbon molecules, organic molecules that you and I exist on. Nitrogen is the same idea. Nitrogen gas. Our planet is 80% our atmosphere. Nitrogen gas, about 80 to 20, nitrogen and oxygen, with some other materials that are in there. Without that nitrogen, we have to, bacteria take the nitrogen out of the atmosphere and fix it. It isn't fix in a sense of repair. It's fix in a sense an affix, stapling molecules together. So we take those other carbon fixed molecules and add nitrogen. Without them, we wouldn't have amino acids, which make proteins, and we wouldn't have nucleotides, which make DNA and RNA. That's why they're so important. There's nothing more. We have more bacteria than any kind of organisms. We have lots of viruses, and we have them living closely in associated, as you can see, in a system and obtaining nutrients and water, protecting from disease. So there's a whole interplay. Maybe we ought to stop it here without me going into another tangent about, things that impact the planet on an ecological basis. So we'll stop here. All good? Like I said, I record these and I will distribute them to you. I will either incorporate it as YouTubes or send them to you, by way of our, Blackboard site.